Hello, my name is Eddie Collins, and this is my photosynthesis video. I will be explaining how DNA ultimately ends up making amino acids. There are two phases to protein synthesis. First is the transcription. The DNA whose shape is naturally double helix breaks apart due to helios. The DNA is then split into two strands. A complementary strand paired up with one half and the other half is copied down to a mRNA, a messenger ribonucleic acid. The DNA then returns to a naturally back to a double helix with the help of ligase. The messenger RNA then goes outside the nucleus and then attaches to a smaller part of a ribosome. While the smaller part is attached to an mRNA, the bigger half of the ribosome clamps down onto the smaller part. This phase is called translation, as the mRNA is translating the nitrogen base's information so that it can be copied down to codons. And the codons are free sets of nitrogen bases that the transport RNA uses to find the correct amino acids to bring. The amino acids tRNA then carries bonds up together, and then the tRNA moves away from the strand, from the linked amino acids. This stage is called translation, as the mRNA is taking the DNA nitrogen bases and translated into an mRNA code so that tRNA can read it and match it up with its anticodons. Codons are free pairs of nitrogen bases, as again. Nitrogen bases are bonded together by either thymine to adenine or cytose to guanine, but in mRNA translation, the thymine is translated to uracil,